Oh, thank you very much, and thank you, Gerald, for a very, very good presentation. I guess I'll start by commending you for doing this project. It's a very important public health problem, as you very clearly stated. I mean, both because HIV is such an important public health problem, especially in children, and also because it is so poorly understood in Africans, which is where the burden of disease is greatest. And I also want to commend you for the really strong training you've undertaken through the H3 Africa program and being able to bring this training to Africa. Because as you kind of pointed out, a lot of this training can happen there. There's no reason why you don't need machinery to run whole exome sequencing and all that stuff. The more important thing is how to analyze that data, which is very challenging, as I'm sure you've learned. And so I think that's very commendable that you've already begun this training in Kampala. So with that, can I start some questions?
Yes, please go ahead.
Okay, so I've always found it interesting, and maybe through your work you've come to think about this. Why is it that the loci that are associated with HIV progression in Caucasian populations don't seem to matter? They're not associated in African populations. Why is it that the genetic architecture in African individuals is so different?
Can I answer that first?
Yes. Yes.
So what I would think is because looking at the migration events and Africa being the home for the modern humans, probably the Caucasians, by the time they left, they went to a new environment where they acquired different genetic architecture that has been shaped for so many years by different environmental factors. Then when it comes down to the African continent, we have individuals who are in a totally different environmental setting, which is shaped by totally different environmental factors. So probably why we cannot replicate the signals that are present among the Caucasians is because the environments have shaped these two populations in very different directions. So what happens there may not easily be replicated here. And what we can know is the variation among the Caucasians is just a subset of the variation among the African population. So it's just that the variation there is just a small numerator of a big denominator. So that's why it is important to come and probe the denominator, which is down here on the continent. So I think that's maybe the reason why we can't replicate what we have there, because it's a small sub-component of the variation here.
I think the way you put that in terms of the denominator is actually a very good way to look at it. So, and a related question is this, because you mentioned HIV, the genetic variation in the virus.
Yeah.
How does that play into the same question? Because I can't actually remember what the genetic variation of the virus looks like in Caucasian populations versus Africa.
Okay. Thank you. So what I can say here is we did not collect this variation among the HIV strains that were infecting the pediatric population, but what we know in Uganda here, we have sub-strains of HIV. And the common HIV here is HIV-1 that probably infects close to over 70% of the individuals here. So we consider that as not likely to affect the population, but I can say here is that's a limitation for this analysis I'm presenting.
But at the same time, I think it's a future direction to see how does genetic variation in the virus synthesize or synergize rather with the genetic variation in the human host.
Yeah.
But I also think, and this is sort of an aside, I think, because I do, as Professor Moses mentioned, I do mostly TB genetics. And I think that a lot of the reason why we see different results for human genetic associations in different world populations is because of the background of the mycobacterial genetics.
Yeah.
Because there's global diversity in the pathogen and global diversity in the human, and those two things interact in some way.
Yeah.
But that's just sort of an aside. I wonder if that happens in HIV as well.
I think it does. It does. But of course, what here you can look at is HIV is a small genome of about 10 kb, and TB is about 4.4 million base pairs. So comparing the two sizes, how much variation are you likely to find in a 10 kb genome versus 4.4 million base pairs? So in TB, I think the implication of the variation in the pathogen versus the variation in the host, looking at those two, I think the effect turns out to be more pronounced in the bigger genome like TB as compared to a smaller genome of 10 kb in HIV. But that is very important.
So thank you for that. So I'm mostly going through my questions in order of your slides. I think I might go off topic at some point, but could you? I just first to say I think the extreme phenotype design is, I think it's very powerful, and I think for the study of HIV, I think it's very relevant, and I think it's the right way to do it. And I think your pediatric study design is actually a really nice way to see that, because you have, like you said in your paper, you know the point at which HIV exposure happens,
Excellent.
which doesn't happen with lots of other pathogens. And so you know the point at which exposure happened, and then you can time how long it takes to progress. So just a question about your study populations. What is the age range? Do you know the age range of your rapid progressors versus your long-term progressors?
The age range for the rapid progressors, the average was about less than 50 months. Let me see. I have it here. So here I have the time to progression. So on the graph I have on the right here, the average time to progression for the rapid progressors was less than, I think this is less than 20 months. That's the average for the rapid progressors. Then for the long-term non-progressors, the average was about 160 months.
Are you, so is that, I'm trying to find the slide.
That is at the
No,
point,
here.
yes, at the point, that is slide number 31. So that is at the point when the diagnosis was made. So that was the average.
Okay.
Yeah, 31.
So what is the maximum age of the long-term non-progressors? Is that about 250 months? Is that right?
The maximum was 250 months. That's the maximum. I think there are about some 6 individuals who are about 250 months.
So those are older.
Yeah.
Those are like older adult, like young adults, correct?
Yeah.
Yeah. Is it? So is it possible that some of those individuals got re-exposed?
I would say that's a standard limitation, because you can have adolescents who are sexually active right here, so it's very hard to say. It's very hard to say, but it should be a standard limitation, because...
So yeah, so I wonder how... Okay. Yeah, I mean, and we have this question in TB as well, that we don't know if additional exposures are, is it, once you've been exposed a number of times, is that what finally makes you progress? And so, yeah, that's and I'm looking at your sample sizes again. Because I was going to ask as a follow-up question, how might you evaluate how much of an impact that, having those older individuals in your sample? I mean, on one hand, the contrast is important. You're comparing rapid to long-term non-progressors, and that's your main comparison. So in one sense, it doesn't matter that the long-term non-progressors are, young adults, but at the same time, how would you evaluate what impact that has?
Okay, so I would think it would require typing the virus, but that's a little bit complex, I think. Teasing out the variation in the subtypes. Probably if there are individuals who have either recombinant strains or have multiple infections of the HIV strain, that will be the way to look at that. And then you're able to see, are they the ones contributing to the phenotype of LTNP? Are you there?
Yes, I'm just writing things down.
Okay.
I hadn't thought of it that way. That's a good way to do it. Okay, so now my next question is about slide 33.
Okay.
And this is less a question for you and how, like, your analysis or anything about your project. This is a question about the chip.
Yes.
I'm struck by the fact that this is an exome sequencing platform, correct?
Yes.
And so it's exome, but only 12.4% of your variants were exons.
Yeah.
Why is that?
Okay, so what I can talk about the capture kit, which is called Vikrom version 2.1 then, because these were run on using two capture kits. These are being developed by the Baylor Human Genome Sequencing Center, and they are known from Agilent, maybe Nebogen, the known manufacturer of these exome capture kits. So how they are developed is, I think it captures close to 44 million base pairs of the exome variation. And to me, probably this is the limitation, that the capture kit itself has been developed using sequence variation from the Caucasian populations. So maybe in the long term, probably they need to use African genomes to develop a capture kit that can address this. And like you said, 12% is so small a capture percentage when we compare. I believe if you did the same study among the Caucasian, what you capture as exonic will be more than 12% because the kit was developed using so many genomes that have been sequenced among the Caucasian populations. But I think of late this will change because now we have so many African genomes that have been dumped in public databases. So if Baylor is looking at having business in the next few years, they may need to develop an African specific capture kit for exome variation.
That's a very interesting point. So, two follow-up questions. One, do you think whole genome sequencing, although I know it's more expensive, would be better in this context?
Excellent, it would be better. I believe we had the monies, we would have done it whole genome sequencing in this context.
Yeah. That's for sure. And this is a sort of a why I'm asking sort of question, because I've seen the same sort of thing happen with GWAS chips, right? So chips that are just designed with SNPs that are supposed to be exon rich and end up looking very much like your data. And so do you think that's another example of, and that chip actually happened to be analyzed in a Tanzanian and Ugandan population. So do you think that's the same thing where the chips are poorly designed because they were designed based on Caucasian genetic variation and not African genetic variation?
It would still be true because most of the chips that have been developed have been they use the Caucasian genomes to develop them. But still, like I said in one of the recommendations slides, the H3 Africa has developed a customized African chip which is covering lots of variation from different African populations. And they are making it a little bit cheaper, so $50 per sample, per human sample for typing would be a little bit cheaper. But of course, like what I can say still, if one has the money, you go for whole genome, not even cheap in this way.
Yes, yeah. Okay, I'm gonna, actually, that actually transitions well to my next question. So I think, let me see, what slide was it? 36? Make sure. Yeah. So slide 36 is very interesting because you show the number of, it compares the variants that you saw in Uganda versus Botswana.
Yes.
And how many of those were common in the combined cohort.
Yeah.
And I think this is so important for study design and how you interpret, because your goal in studying the genetics of HIV is, like you said at the very beginning, you want to design a better, you want to understand the biology so you can design a better vaccine or do risk assessment or all these things. But there's a large amount of the variation that is common, but there's so much that is unique.
Yep.
So how does that affect both your design of future studies and interpretation?
Okay, so like still looking at that, the rare variation, which is equally big here, is I think they still need to do some more representation of the different small tribes that are contributing to this variation. Because what you can see from this, the rare variation, some of them were from individuals who are from a tribe that is represented here in this sample size by maybe 5 individuals. So if you have 5 individuals representing a tribe here, still that rare variation cannot be captured even if you have to do SNP genotyping later. So what I suggest here is, even before going forward, we still need to do lots of sequencing in even the smallest tribes here, and then have their variation captured in the big databases. Then subsequently this can be meaningful in the end. But for now, the rare variation is big, which you wouldn't get among Caucasians. You can't have these many rare variants if you are to do exome sequencing among a Caucasian population.
So do you think that the unique variants that you find, because as you said, you're going to find, because of smaller ethnic subsets, you're going to, you'll have unique genetic variants. Do you think those are clinically and biologically just as important, the unique variants that are unique to Uganda or unique to Botswana than the ones you find, compared to the ones you find that are common?
Yeah, I think they are unique, and in the way there is some, I was looking at doing some classification, looking at the variant classes for these rare variants, and trying to check them against the databases such as the HGMD and see, are they also there? In case they are there, have they been associated with the disease causing, have they been associated with the disease? But what I can say for now is the rare variants that are captured here, many of them were missense, like you have seen. The distribution of the missense variants were so many, and I think they have clinical relevance. That's what I can say. The only problem is we do not have the clinical data regarding these patients. If we knew much of what their clinical data was, then we can probably associate them with what we are seeing clinically among these patients, beyond using it as predicting LTNP status or rapid progression status. I don't know if I've tried to answer the question you asked.
No, you did. I think a lot of, but I think you also made a good point that there's a strong motivation to do additional research in additional larger populations, and to but you pointed out too that understanding the functional implications of these variants is very important. And I think you did an excellent job of going into all the different bioinformatic databases to do that, because that's not easy. It's pretty hard to do.
Thank you.
Okay. Go ahead.
Okay. So I was just appreciating your comment.
Yeah, well, and I should have prefaced all of this by saying that, I mean, having studied, TB in Africa for quite a while, I mean, my whole life, I'm very appreciative of this kind of analysis because it needs to be done because the genetic variation in Africa is so different and there's just so much to be learned. And there's a very nice paper that was recently published in Cell by a couple of my colleagues, pointing out that there is a great need to do additional research in Africa. So you're a little bit, preaching to the choir on this one. So my next question is, so in your first analysis, you were looking genome-wide. In your second analysis, you focused on specific chemokine and cytokine gene regions.
Yes.
And so I'm curious about the ordering of that process because on one hand, you would think that if you found something at the genome-wide level, if it was important, like those chemokine genes could have come up in your first aid. But on the other hand, we know that one of the most important, susceptibility genes for malaria has never come up in a genome-wide search. I just have a question. My question, I'll pose it this way, is about the philosophy of doing the genome-wide analysis first and then following it with the chemokine and cytokine gene-centric approach.
Okay, so thanks for that question. So what happened is, I think there are about 2 papers, one in PNAS that confirms that much of the variation that has been discovered among the Caucasian that has been responsible for long terminal progression is common variation. That means, I think about 3 papers confirmed that it's common variation that is responsible for disease LTNP status. So that's the rationale for first trying to analyze this data set at common variation level. We thought in case we had, whether it was a chemokine or their ligand, as the one responsible for this phenotype, it would come up in the common variation analysis. And none of So we said, since this is an exome phenotype design and HIV progression being a complex phenotype, we may need to find a few individuals who have rare variants that may be damaging and enriched and probably able to be responsible for these phenotypes. So the rationale was, look for the common variants. If you can't see it, then go down, look at chemokines and their ligands, because most of the studies that have been done have identified these at different levels. So that formed the rationale of doing a third objective, which was specifically interrogating on the chemokines and their ligands.
Now, and then my other question is, I noticed that you did gene-based analyses.
Yes.
Did those analyses reveal anything different than the the individual variant analyses?
So for the chemokine analysis, we first still had to run the single outside test association testing using the fish exact test. And as expected, we don't have any hits that were statistically significant because we had to do multiple testing and everything else disappeared. All the p-values that we had that were less than 0.05, I think it is part of the supplemental in the paper, all of them disappeared after correcting for multiple testing. But then CCR6 attained a nominal statistical significance and it had only 4 mutations that were of interest in there, and however, that nominal statistical significance collapsed after correcting for multiple testing. So that was it.
Okay.
Yeah.
So can I ask you then about the multiple testing correction and how did you determine the number of tests to correct for?
It was the number of genes that were in that data set, in the gene-based analysis.
Okay, thanks. And so that was for the chemokine cytokine analysis,
Yes.
gene analysis, correct?
Yes.
So you corrected for the gene okay, then for the single variant analysis, what was your correction based on?
It was based on for the common variants, we had 314 individuals. So we corrected that using the standard cutoff for exome-wide significance, which is 2.5 times 10 power minus 6. And how that is derived is they take the standard p-value of 0.05 and then divide it by the number of genes in a human being, which is about 20,000 genes, and they come up with 2.5 times 10 power minus 6 as the cutoff. So anything that would be less than that was to be taken to the next level. So that's how we corrected for the multiple testing at the common variant.
And the common variant, okay.
Yes.
Because I thought in your paper, so, and I guess I'm going back to the prior, I don't remember what name that was, when you did the genome-wide analysis.
Yeah.
I thought it said that your correction was based on the number of missense variants, not total variants. Am I correct?
No, that's not correct.
Okay.
Yeah.
All right, so it was just based on the number of genes, not the number of variants.
So the cutoff for the common variant, we use the standard cutoff for exome-wide significance, which is, the threshold is 2.5 times 10 power minus 6. So anything that was below that could be...
Yeah.
I
don't know why I thought it should be more strict than that. I thought it was, usually the standard cutoff was really tiny.
No, that is the most stringent among, when you're dealing that with the calculation at the exome-wide significance. But if it is genome-wide significance, then it is, it is power minus 8, which is corrected at...
Hey, that's your lot.
I missed that, sorry?
No, I'm just going back over my notes to make sure if I had any other questions.
Okay.
So, in your, the journal Human Genetics,
Yes.
where you compared the frequency of different variants in Ugandans and Botswana
Yes.
to what's available in public databases.
Yeah.
Do you think there's any bias because that you have is ascertained through affected children or maybe, yes, affected children?
Kat, I missed that, sorry, come again. Hello?
Can you hear me?
No, I missed that.
Okay, so in the paper, in the American Journal of Human Genetics, and I forget which objective that was in your dissertation.
Yes.
You compared the frequency of different genetic variants in your Uganda and Botswana populations to public databases.
Yes, yes.
Do you think that you may have seen an increased frequency of different specific variants because your population was HIV exposed or, had progressed to HIV/AIDS?
I wouldn't think that is true because HIV has been around for maybe coming to 30 years, and I don't think it has impacted so much that we have a different genetic variation in this generation that is missing in the bigger database because of HIV. So I don't think it has impacted to introduce a variation that is very much unique and is missing in the database because of that. I wouldn't think so.
Okay. I have one more last question. I'm trying to figure out how to formulate it because I noticed that you studied a bit on ethics, and that's one thing I'm very interested in. Given how recently, now that it's becoming more common to train African scientists in genetics and bioinformatics, it would be really, important for, everyone, all of those scientists to get papers and grants and all that stuff. But at the same time, there's this push to publicly release genetic data. In which case, other places of, U.S. or UK-based institutions could maybe publish on them more quickly, or more importantly, they might not, depending on the scientist, you don't know how they would acknowledge the contribution of the people who originally generated and collected that data. So what is your perspective on genetic data sharing in terms of both like the sensitivity to genetics in Africa and then also a recognition of African scientists?
Okay, that's a nice question. So what I can say is for under the H3 Africa, the data set has, I think for now, the H3 Africa is trying to come up with a curfew on how much time they have to hold on to the data before it goes into the public database. I would say yes and no, it would be right, but what I know, if you dump these data sets in the public databases, within the next 2 or 3 years, you will have scientists from the Western world discovering so many things from this data set, and they will never make any contribution or acknowledgement of the African scientists. This is the way things are being done, and that's why the H3 Africa is still holding on to this data set. For some time, so they allow African scientists. I should say maybe we are trying to move at a slower pace compared to the Western scientists, but that is being negotiated at the H3 Africa level, and they will be coming up with a time frame during which now they will say, whoever has this data set under the H3 Africa, you have to dump it in the public repository. But for now, I think we have a priority to use it and make whichever discoveries we can, but I think it won't be like that for long.
Okay.
Yeah.
That's a good answer.
Okay.
I think that actually covers all the questions I had.
Okay, thank you.